All right, good afternoon. This is the last session for this year's annual meeting of the Marine Mammal Commission, and of course we saved the best for last, the Arctic session. And without a doubt, it's the most marine mammal diverse of all the sessions. So we have a talk on polar bears, on ringed seals, right whales, and beluga whales. So we cover almost all of the different groups of marine mammals. This is mostly just presentations to update the commission. And so there's not a lot of time available for questions and discussion. Unless speakers finish early, then we'll certainly have some time. Our first talk today will be Eric Regier, and Eric is a research biologist here at the University of Washington, but he also has lots of experience working at Fish and Wildlife Service and USGS before that. And so Eric is going to talk to us about Chukchi Sea polar bears.
Okay, great. Thank you, Robert, and thank you for the opportunity to be here. Again, my name is Eric Regier. I'm a research scientist here at the Polar Science Center at the University of Washington, and I'll be providing today a scientific update for polar bears in the Chukchi Sea region. So here's an overview of the topics I'll cover. First, I'll present a current and in its final stages effort to estimate demographic parameters for the Chukchi Sea subpopulation from capture-recapture data collected in American territory over the past 10 years. I'll talk about an in-progress harvest risk assessment for the population, briefly touch on instrument-based aerial surveys led by the National Marine Fisheries Service, a new effort to delineate the biological boundary, biological population delineation between the Chukchi Sea and Southern Beaufort Sea subpopulations, and finally a brief note on a promising new collaboration between the American and Russian scientists on Wrangel Island. So first, talking about the demographic parameter estimation. Some people here are familiar. The United States Fish and Wildlife Service and its partners performed capture-recapture research off the west coast of Alaska from 2008 to 2016. This actually continued in 2017, although those data aren't part of the analysis that I'll talk about. During this effort, about 420 polar bears were captured, studied, and released on the sea ice west of Alaska in the spring. 107 radio collars were deployed. These were GPS devices that stayed on the animal for 1 year and then were released by an automatic release device, and also 77 prototype satellite tags. We were really trying to get away from the more cumbersome radio collars and have success with smaller ear-mounted tags on the polar bears. This study broadly had several lines of investigation. First, looking at body condition, reproduction, health, and nutritional ecology. Second, trying to get a handle on habitat use and population delineation. And third, try to estimate abundance and vital rates for the bears. And this third step, abundance and vital rate estimation, really built on the first two lines of investigation there. Like everything I'll talk about here today, this is a collaborative effort with a lot of different partners. So here, just give you a sense of where this research occurred. The left panel zoomed out. The black area there between the Seward and Lisburn Peninsulas is where this work took place in the spring of the year. Right panel there are the capture locations for these bears out on the sea ice. As far as an analytical framework, we did some really exciting stuff. We analyzed these data with an integrated population model. I'm sure some of you are familiar with these approaches, and it had several advantages compared to previous approaches to analyze capture-recapture data. We were able to make efficient use of multiple data types, bring in capture-recapture data, radiotelemetry data, and count data all into the same framework. These IPMs have advantages. They can link demographic rates to population processes, such that if you estimate a survival rate, that rate is consistent with your estimate of population growth rate from the model. One of our real goals here was to mitigate some of the biases that are common in capture-recapture studies for polar bears. There's a long history of doing these studies in the spring on the sea ice and coming out with especially estimates of survival that have a meaningful bias, and that's related to movement of animals on the sea ice platform in and out of often a relatively small, a relatively geographically restricted study area. And this IPM framework was also implemented in a Bayesian framework, which gave us the ability to incorporate auxiliary information as informative priors. And we actually did draw both on other scientific studies and on traditional ecological knowledge to inform some of the parameters in the model. So very briefly, with the caveat that these results are our draft material, pre-decisional, we were able to estimate sex and age-specific survival rates. I think we took big steps toward mitigating the bias that you often see in survival. There still probably was some, as evidenced by our goodness of fit testing, but we took some steps forward there. A little more straightforward to interpret, we estimated reproductive parameters. We found that breeding probability and cub of the year, that's first year cub survival, were about average for the species. Yearling survival and cub of the year litter sizes were high, so some of these reproductive metrics were quite high. And an interesting one was we also looked at the number of yearlings per adult female. That's a metric that integrates both breeding probability and first year survival, so it's a good index of recruitment. And we actually had historic data from the late 80s and early 90s to compare to, and found that our values currently are very similar to what they were in the late 80s and early 90s, and also that we didn't see a trend over our 8-year study, 2008 to 2016. So some evidence there that the productivity of bears in this part of the world hasn't declined or changed significantly in the past 20, 30 years. We were able to estimate density within this relatively limited study area, about 3 bears per 1,000 kilometers squared. Somewhat anecdotally, that's very similar to the estimate of density from a pilot aerial survey performed in the same region in the late 1980s. We did derive an estimate of abundance for this larger Chukchi area, and we did that by extrapolating densities from the study area based on relationships between animal density and a spatially explicit habitat quality metric. When we did that, the estimate of abundance within the Chukchi subpopulation boundary as defined by the Polar Bear Specialist Group was about 2,900 bears. Importantly, this Chukchi subpopulation boundary is different than the population boundary for the Alaska Chukotka population as defined under a treaty between the U.S. and Russia. They overlap, but that Alaska Chukotka population boundary is larger. We did not take the next step of estimating abundance for that larger area because of concerns of extrapolating too far beyond the actual location in which our data were collected. So some key messages. First, empirical estimates of population size and vital rates for bears in this part of the world. Our findings suggest that the population was productive during this period. It's consistent with several other lines of scientific evidence and evidence from traditional and local ecological knowledge. Similar to most polar bear subpopulations, the future is uncertain. These animals are spending about a month longer on shore every year because of sea ice loss. More animals are coming on shore. At some point, one would anticipate that there's a nutritional threshold that these animals will cross and start to have some negative demographic effects, but we did not find evidence for that from 2008 to 2016. Typical issues for polar bears, large statistical uncertainty, potential bias in some parameters. At the end of the day, though, I think we developed some information, quantitative information, that is urgently needed for management and conservation of these bears. So this work is currently in review in the Journal of Scientific Reports. Clearly a product of many collaborators and a lot of individuals and agencies. So one of the things we want to do with these new estimates of demographic parameters is perform a quantitative harvest risk assessment. We want to try to evaluate demography based on these parameters. What do these estimates of survival, reproduction, et cetera, mean for the subpopulation? What can we determine about population status? And so this is in progress now. Just talk very briefly about the methods, but we're evaluating the biological risk associated with a wide range of harvest strategies. And the goal here is to provide this assessment to the U.S.-Russia Polar Bear Commission, who's responsible for management and conservation decisions for these bears, to inform their determination of sustainable harvest level, which is related to the potential annual taking limits on the population as far as a sustainable harvest. Our assessment uses a coupled modeling and management framework. This is a science-based approach to trying to balance the population effects of harvest with maintaining continued opportunities for subsistence use, clearly recognizing the cultural, nutritional, and maybe not so much in the U.S., but in other parts of the Arctic, the economic importance of subsistence harvest. This work was published last year in the Journal of Applied Ecology. One of the motivations for this framework was the question about how to harvest polar bear populations that are facing current or future declines due to habitat loss. Very briefly, at the heart of this framework is a matrix-based population model. Our goal here was to provide a faithful representation of the life cycle of the polar bear, extended reproductive cycle, importance of sex and age-specific vital rates. A key development was that we developed a density-dependent model based both on population theory and on some empirical evidence for other subpopulations. Basically, this is an attempt to describe how polar bear populations respond when it gets crowded. Having a plausible model of density dependence is very important if we're going to try to use this framework to evaluate the concurrent effects of both habitat loss, changes in carrying capacity, and direct human-caused removals due to harvest. So some of the key messages for this, critically, this is a state-dependent management approach. That means management is dependent on current conditions. It provides a direct link between research and management. And so how this works is every X number of years, 5, 10, 15 years, there'll be new population studies. Estimates of intrinsic growth rate from the new population study will go to inform the harvest rate. Estimates of abundance from the new population study will go to inform the harvest level. And so clearly when we're talking about populations that are facing habitat change, that may be facing both density-dependent effects, lowering carrying capacity, potentially density-independent effects, reduced intrinsic growth rate, having this type of state-dependent framework is very important. This is the only modeling framework available for polar bears that considers density dependence, management interval, that's the number of years elapsed between new studies and updates to management. The quality, the precision of the population data. If the data are less certain, things are riskier. And also, importantly, the potential effects of habitat loss due to climate change. Again, our goal to provide the managers with the probabilities of meeting management objectives that are consistent with the definition of sustainable harvest level in the U.S.-Russia agreement for a wide range of harvest strategies that vary in terms of those things that authorizing agencies can often control: the harvest level, the harvest rate, the harvest composition, et cetera. Another final point is that this is an ongoing effort, and we've made it a collaborative process involving American and Russian members of the scientific working group that is formed to advise the commissioners of this U.S.-Russia agreement. And so we have a wide range of backgrounds and expertise on this, from federal scientists, North Slope Borough, ADF&G, and people with traditional and local ecological knowledge. So very briefly, also touch on the Chukchi and East Siberian surveys for bearded seals, ring seals, and polar bears. Many of you are familiar with this effort. It's led by the National Marine Fisheries Service, Peter Boving and Aaron Moreland. Field work was completed in 2016. There were coordinated instrument-based aerial surveys on both the U.S. side and on the Russian side. A very, very large effort, 25 flights on the U.S. side, about 16,000 kilometers surveyed. Both surveys in the U.S. and Russia made use of joint thermal cameras and high-resolution color cameras, and it provided imagery like this. So on the left side is the high-res color image, on the right side is the thermal image. On this one, you can maybe see the polar bear in both, and you can zoom in here and see it there. Other instances, rough sea ice, much harder to see the polar bear on the left side, but again, zooming in, you see that there is indeed an animal there. So a few points about this. The survey design was based on statistical modeling to determine the appropriate level of survey effort. Coverage of the actual survey exceeded this target. However, the measured detection of polar bears was lower than anticipated, and indeed only 3 bears were detected on effort on the U.S. side. There are a different number of detections on the Russian side. So NIMS and collaborators are actively working on efforts to improve the detection, including developing new algorithms using both the thermal and the color data sets, considering expanding the spectral range, looking at UV to eliminate the potential effect of behavior on visual and thermal detection, and hoping to test some of these new algorithms in the future, and also conduct some additional survey flights to develop the methodology. And again, please follow up with Aaron Moreland or Peter Boving with questions. Slide or two on the effort to update population delineation between the Chukchi Sea and the southern Beaufort Sea subpopulations of polar bears. This is an effort being led out of the Colorado State University and by Dr. Ryan Wilson with the U.S. Fish and Wildlife Service in Alaska, also collaborators from the U.S. Geological Survey, George Durner and Todd Atwood. What they've done here is developed an updated methodology to try to delineate biological populations. It's within a Bayesian hierarchical framework. This methodology has several advantages compared to what was previously available. For example, it accounts for location error. It allows the inclusion of all location data. Importantly, it accounts for sea ice movements in estimating population membership in the boundaries. These polar bears are out there on a very dynamic habitat. We'll do research in the spring and land with the helicopter and turn the GPS off and turn it back on an hour later and wonder if something's broken, and it's not. It's just that you drifted 8 miles. So this framework takes that into consideration. Estimate delineation and boundaries jointly with data from 2 subpopulations, probability of subpopulation membership, and an estimate of uncertainty in the boundary. So here's, again, preliminary results. These are pre-decisional. This work is currently in review at the Journal of Biometrics. The solid line there on the right side is the preliminary estimate of the point of delineation between the southern Beaufort Sea subpopulation north of Alaska, Chukchi Sea west of Alaska. As you can see, that line runs right about through Wainwright. The dashed line is uncertainty interval, which goes about from Icy Cape to just east of Point Barrow. So importantly, there's not a direct link between this work and some management decision, for example, regarding a boundary. This is an analysis of population delineation intended to inform potential management decisions. And the last slide or two is the study I mentioned, which is polar bear studies on Wrangell Island. Looking again at the same map, you see Wrangell Island is located there, kind of on the left, north of Chukotka. Wrangell is really the ecological heart of this polar bear subpopulation. The majority of the subpopulation builds their maternity dens there and gives birth on Wrangell Island. A lot of bears use Wrangell Island as a resting platform in the summer when the sea ice recedes into the polar basin. Bears will come on shore, spend 2, 3, 4 months on Wrangell. It's a Zapovednik, which means a strict nature reserve. It's a protected area, so the bears can spend time there without human disturbance. This is the first on-the-ground collaboration between American and Russian scientists on polar bears in several decades. We're very happy about this. It took multiple years to get off the ground. Our study methods involve ground-based observational surveys, counting the number of bears, distribution, their habitat use, body condition, that means their fatness, their behavior, reproductive indices, and composition, sex and age composition. And we're also developing some non-invasive genetic sampling methods of the bears on this island. There's a captive audience there for a few months, so it's a great opportunity to collect genetic samples without physical capture or chemical immobilization. Our goals are really to understand the ecology of the subpopulation during the ice retreat season, during the summer and the autumn. Again, most of the U.S. research has been in the spring on the sea ice, and so as far as the annual cycle, there's kind of a missing piece there that we haven't had direct data on previously. Clearly, we're also trying to learn things about population demography from this study. So we successfully completed field work in 2016 and 17. 179 bears observed on survey in 16, 589 in 2017. So that left map there shows the route of the ground-based observational surveys. That's about 1,000 kilometers that was covered in tundra vehicles and ATVs while making observations of bears. The right side is one of these hair snare traps. This is an approach that was developed by North Slope Burrow and ADF&G and that we're trying out there. But in this box, there are some little brushes that when the bear puts a paw or a head into the box, these little like test tube brushes, pull out a hair or two. There's no food in there. There's just a few drops of kind of stinky fish water to serve as an attractant. So the study's anticipated to continue this year and for several more. And again, this is a collaborative effort between the Wrangell Island State Nature Reserve, University of Washington, critically the Fish and Wildlife Service, the Russian Ministry of Natural Resources and the Ecology, and hopefully also with the Borough and Alaska Department of Fish and Game on board going into future years. Just one more picture because it was fun. On the west coast of Wrangell last year, there was 189 bears within a kilometer of a bowhead whale carcass. And so this is actually a picture. A tourist ship had come by at the same time. This was kind of all over the internet or in some parts of the internet, and there were some professional photographers that got this picture from a skiff. But it was just a hill with polar bears like sheep on it. It was the most amazing thing. But it does speak to the point that there are fairly reliable sources of large whale carcasses, gray whales and bowhead whales, that because of circulation patterns tend to wash up on Wrangell, tend to wash up on the Chilcotin coast. I think there's a new paper coming out looking at that, but we can anticipate this being an increasingly important food source, I think, for these bears in future years as the sea ice continues to diminish. So with that, it says 22 seconds. I know I talked fast, but thank you all very much.
Thank you very much, Eric, to you and to Ryan and to the NIMS folks, that all of these data are much improvement over what we've had in the past for Chukchi sea bears, and I look forward to seeing continued work. We don't have time for questions, and so our next speaker will be Amy Lang, and she is talking about stock structure in ring seals, and this project was funded or partially funded by the Marine Mammal Commission.
Thank you, Robert. So today I'm going to give a brief update on some work we have in progress to better understand population structure in Arctic ring seals. This work is ongoing, so the results I'll present today represent essentially just a preview of what we found so far, and we hope to have more complete results in the near future. But before I get started, I just wanted to acknowledge my collaborators and some of the sample contributors for this project. And I won't go through the names in the interest of time, but in particular, I wanted to point out that this project wouldn't have been possible without the samples that have been collected by the Alaska Department of Fish and Game's Ice Seal Biomonitoring Program, where they work with villages to collect samples from seals that are taken in the subsistence harvest. So 5 subspecies of ring seals are currently recognized. This includes the Arctic ring seal, which is the most widespread, the Okhotsk ring seal in the western North Pacific, the Baltic ring seal in the Baltic Sea, and then 2 subspecies of freshwater seals, one that inhabits Lake Ladoga and one that inhabits Lake Sami in southeastern Finland. Now, within the Arctic, ring seals are widely distributed and abundant. Combined regional estimates covering a large portion of their range suggest an abundance of around 3 million animals, and estimates of the number of seals using the Alaskan Chukchi and Beaufort Seas suggest that these areas contain around 300,000 seals. And I should mention here, and Eric mentioned this as well, that there are efforts underway by the Alaska Fisheries Science Center to get a more comprehensive abundance estimate for this species. Within U.S. waters, a single Alaska stock is recognized. Whoops. And that stock is co-managed by the National Marine Fisheries Service and the Alaska Native organization, the Ice Seal Committee. So ring seals rely on sea ice and snow cover for survival and reproduction. And as we're all aware, over the past several decades, the extent, thickness, and seasonal duration of Arctic sea ice has declined and is continuing to decline due to climate warming, which has raised concern for these seals. Declines in body condition, reproductive rates, pup survival, and foraging behavior have been reported in ring seals in some areas. The reduction of sea ice in the Arctic has not been geographically uniform, which suggests that some parts of the Arctic ring seal's range may be more impacted than others. This highlights the need to better understand population structure in these seals, because it could impact how the seals respond to continued climate warming. If these seals are panmictic throughout their range, then seals that have previously used areas which now are ice-free would be able to then just move to other areas where suitable habitat remains. However, if the seals show natal philopatry such that they tend to return to breed in the same area where they were born, then localized depletion of seals in these areas could occur. And that could potentially result in the loss of genetic diversity important to the evolutionary potential of the species. Now, most of what we currently know about population structure in Arctic ring seals comes from telemetry and genetic studies. The tagging studies have shown that seals range widely during the foraging season, but that their movements are more restricted during winter-spring months. Perhaps most relevant to the population structure question, many of the seals whose tags have remained attached for a full year have shown that they return to the same wintering area, which is where they breed, in subsequent years. However, it's not clear if such return represents return to natal areas or return based on other factors, such as knowledge of high-quality breeding habitat. 2 genetic studies have been conducted thus far to look at population structure in Arctic ring seals, and these studies have failed to detect population structure throughout most of the range. These studies have relied on what I'll call traditional genetic markers, including mitochondrial DNA sequence data and a small number of nuclear microsatellite markers. Although the results of these genetic studies are consistent with a lack of population structure in ring seals, they have not been able to rule out the possibility that more subtle genetic structure exists, but was not detected using the samples and genetic markers that were analyzed. We know that genetic differences accumulate slowly in very large populations, and traditional markers can have little power to detect low, but potentially biologically significant levels of differentiation. Recent technological advances, what we often refer to as next-generation sequencing, now allow large numbers of SNPs to be genotyped in a cost-effective way and without requiring that genome data be available. And those extensive data sets have been shown to substantially increase the power of genetic analyses, even with limited sample sizes. So for this study, we essentially decided that we need to get a bigger hammer. We took advantage of some of the recent advances in next-generation sequencing and started a project to discover and genotype a large number of SNP loci in Arctic ring seals. I'm not going to go into the details of the methodology here, but we used a method that's referred to as DART-seq to do the genotyping, which for the geneticists in the room is a double digest RAD-seq-like approach. This map shows the distribution of samples analyzed in the study. For this work, we only used samples collected during March through May, which is considered to be the breeding season. And as I mentioned earlier, the majority of samples used in this work were collected as part of the ADFG's biomonitoring program. We then stratified these samples to align as closely as possible with the management areas used by the ICEAL committee, although note that we don't really know what sort of environmental or other factors could lead to population structuring in these seals. So this was just a proxy to start with. So basically, we extracted DNA from these samples, and then we sent them to the DART-seq company, which is in Australia, to do the sequencing. And in the data they sent us back, there were over 100,000 SNP loci that had been identified prior to doing any quality control filtering. However, when we screened the data to identify a reliable and high-quality set of SNP markers, we ended up with a SNP panel of just over 8,000 loci. We also screened the individuals that we had included in the study to determine if any of them were related. And you can see the results in this graph, and I apologize, some of the lines don't show up very well there. But the key point here is that the distribution of relatedness values in our sample set, which is shown in the red peak, were consistent with the individuals being unrelated, with the exception of one outlier pair, which is marked by the red dotted line and which falls near the middle of the distribution expected for half-siblings. Given that these individuals did not share the same mitochondrial DNA, these individuals appear to be paternal half-siblings. Both of them were sampled in Hooper Bay in the spring of 2008, and one of these individuals was estimated, based on the harvest data, to be about 8 months old, while the other was estimated to be about a year old. So that means that they were basically from the same cohort. Although ring seals are considered polygynous, this was a little surprising given that our sample size is so small compared to the abundance. In any case, it does provide evidence of a single male mating with multiple females during a single mating season. And for me, it also highlighted the need to understand more about the movements of seals during their first year, because that affects how you might interpret this information with respect to whether the seals remain in the same area during their first year of life, which seems to be the general understanding for these animals, or if they would migrate with the adults and subsequently return to the area. Now, despite having markedly increased the power to detect genetic differences between groups, our results were largely consistent with those previous studies and showed little to no genetic differentiation between the a priori defined geographic strata. While the magnitude of differentiation was quite low across all the comparisons, there was one comparison that was statistically significant. That was between animals sampled in Hooper Bay and in the Beaufort Sea. And those are the two most geographically disparate areas, and also the areas where we had the highest number of samples. So as I mentioned, this is a work in progress, so we have several other analyses planned. We're currently running a few different clustering type analyses, and those are important because those types of analyses don't require that the samples be stratified into geographic groups beforehand. So it allows us some room to explore different stratification patterns. We're also running analyses to determine if any of the SNP loci included in the study show signs of being under selection, which could allow us to explore adaptive variation in these seals. But what can we say so far? Basically, even though we went out and got a much bigger hammer compared to previous studies, we ended up in a pretty similar place to the earlier studies using more traditional markers. Our results are largely consistent with there being little to no population structure within Arctic ring seals. And this could be good news with respect to climate warming, as it suggests the loss of sea ice habitat may not result in the loss of evolutionary important genetic diversity. However, we did detect some very small differences between the two most geographically distant and most well-sampled strata, which I think needs to be investigated a little more closely before we draw any strong conclusions about the population structure in these seals. In this first step of the project, we chose to increase the power of our analyses to detect genetic differences by substantially increasing the number of loci analyzed. However, this work is based on a relatively small number of samples, particularly when the abundance is considered. So I think it's going to be important in the future that we increase our sample size and fill in some of our sampling gaps to see if any new patterns emerge or if these existing patterns are upheld. And as Robert mentioned, this work was funded by the Marine Mammal Commission, so I want to thank all of you for that, and thank you for your attention.
Thank you very much, Amy. And as with Eric, we don't have time for questions. If folks have questions for either Eric or Amy, please catch them after this session. And our next talk is going to be a tag team here between Jessica and Dana, and I'm not sure Dana is going to go first. And this project was also funded or partially funded by the Marine Mammal Commission as well. So it's all yours.
Thank you. Good afternoon, everyone. Thank you for being here. My name is Dana Wright, and I am going to be speaking about using passive acoustic data to try to unravel the current range of right whales on the Bering Shelf. As was stated, the Marine Mammal Commission personally funded a year of my analysis, so I would like to thank you for that, because it definitely progressed the project. So given this is a public talk, I thought I'd give a little bit of background on the right whale. I think this is a really nice figure from Smith et al. That shows each dot is a sighting or catch of right whales from 1780 through 1920, and you can see that they used to be quite abundant in the North Pacific and Southern Ocean, and that specifically in the North Pacific, it looks like there's kind of two populations, one off of Asia and Russia and one off of North America, which has resulted in kind of an eastern and a western population, and today we're going to be talking about the eastern population. So these are the same data broken into time frames, and I think it really nicely illustrates how efficient whaling was and how in such a short period of time the population was completely decimated. Again, right whales are in red. It's showing some other species as well. And that the latest population estimate is estimates right whale speed about 30 animals for the eastern population, with most of the sightings and detections in the southeastern Bering Sea, which is where we think that the kind of core habitat is. So today, based on a lack of funding, the large potential habitat they used to occupy, and their small population size, there's still a lot we don't know about them, but they're pretty hard to study. So we've utilized passive acoustic monitoring or listening for right whales. So the Marine Mammal Lab from NOAA here in Seattle has deployed recorders called they're called hydrophones, which are underwater microphones that are listening for right whales. So we have recorders at 9 stations in the Bering Shelf and 2 in the Aleutian chain from 2007 to present. And they record on what's known as a duty cycle, or they record for a period of time, shut off, and then turn back on. So we get near continuous sampling, really good resolution to try to find these very rare animals. These recorders are attached to subsurface moorings, so they're fixed. They're listening at one location, and they have about a 20-kilometer range. And the black pentagon in the figure is the right whale critical habitat, which again, it just illustrates kind of how contracted their range is thought to be. So I'm going to start off talking about data on the Bering Shelf from 2011 through 2017. And this is a really interesting time frame. That's the amount of data we've analyzed to date because of how dynamic the Bering Shelf is, for those familiar with the environment. The gray arrows are all the different currents, and I just overlaid red arrows to illustrate that there's a cross shelf and a long shelf kind of flows and a lot going on, making it very dynamic. Also, this area is seasonally covered with sea ice, and it can extend all the way down to the southeastern Bering, depending on the year. So that's obviously going to impact the environment as well as where right whales can go. This is showing some nice oceanographic data. So the top figure is percent ice cover. So I've illustrated or highlighted in yellow the 2011 through 2016 data. And you can see that for the last 3 years, 2014 through 2016, ice did not extend down to M2, and it was also very, very warm. And this is in contrast to the other years where it was cold. And it's thought that these different conditions change the ecosystem to where it might be more favorable prey base for right whales than one of the conditions or the other. So it's just a really natural, interesting data set that we have 2011 through 2017. So in terms of the analysis, we did manual analysis of all data. We didn't subsample. So how that was done was that a human would go through and look at the data in 3-minute time bins and decide if a species was present. For the right whale, we use 2 call types, the up call and the gunshot call. These are the 2 most common call types for all right whale species. And specifically for this population, the gunshot call is actually produced more frequently, which is different than the other populations. So these are both really good markers to try to find this species. So looking at the results, I apologize, these are very small, but I tried to fit as much data as I could. So these are the gunshots in yellow, and so each column is year, 2011 through 2017, and then the rows are going to be the months. So this top row is July. So showing July through October, you can hopefully see that the circles are consistently the biggest in the southeastern Bering in that critical habitat where we thought that they would be. And each circle is the percentage of those 3-minute time bins with calls, with right whale calls out of the time bins sampled per month. And if we overlay our kind of warm, cold time periods, you see some interesting trends that appears 2015, 2016, the range that right whales were heard is a little bit wider on the shelf than in the earlier years, with detections actually north of St. Matthew in 2016, so extending into the northern Bering. If we look at the upcall data, you'll see that the circles are smaller, which is what we thought. They produce that call less frequently, but also the patterns are very similar in that the 2 call types mirrored each other, and that upcalls were also heard in the northern Bering in 2016. And this is not, an unheard of conclusion. There was a handful of catches and sightings from the historical whaling era that support the right whales can be in the northern Bering, but there is contention whether those are misclassified bowhead. But these data seem to support that right whales can occur in the northern Bering. So the question becomes, like, why was there's this difference? And it appears to be, as I alluded to, the oceanography, the base of the food chain. So this is a nice figure showing 2001 through 2017, the cold pool, which is another feature of the Bering Sea. The extent of the cold pool depends on ice and wind. So you can see that in the cold years, it extended into the southeastern Bering, which is when there was ice that also went down to the southeastern Bering. And then in those warm years, the cold pool did not extend that far. And if I just overlay the 2012 and 2016 data from September, you can see that the range of right whales kind of was contracted in the southeastern Bering in 2012, and then in 2016 it was expanded and kind of almost bordered the cold pool. But if we then look at 2017 for that same month, it goes back to being pretty contracted. So these are consistent with the limited tagging data we have of right whales. 2004 was a warm year. You can see the cold pool is just kind of around St. Matthew. And that whale was a lot more active. It moved around a lot more, whereas in 2008 and 2009, those animals had a pretty constricted range. And it's thought that that is, again, due to food availability. And as I alluded to before, we have data back to 2007, so I think it would be, really helpful for data to analyze data back to 2007 to see if it corroborates this hypothesis. Also, briefly, I'd like to touch on the Aleutian Pass data. The Marine Mammal Commission funded a lot of this, and I think there's some pretty interesting results. So we put recorders in the Aleutian Passes because the theory is that right whales will seasonally migrate from the Bering Sea. This is based off of the fact that other right whale species do that, and that there's been rare sightings of right whales off of California and a match re-site off of Hawaii. And it's thought that if they are migrating, that they would likely use the Aleutian Passes, and the one that they would most likely use is Unimak Pass, because it's the first one they would encounter as they leave the critical habitat, and it's also part of the known migratory route of other species like humpback and gray whale. This is, of course, concerning because this is a very high vessel area, and it's also very narrow, about 16 kilometers. So if right whales are there, then there is potential for interaction. So I'd like to call this my bubblegum plot. This is the number of days with right whale calls by month for the two call types, 2009 through 2017. And you can see that there isn't really a pattern, but they are detected at least once in all months except October. So supporting that right whales are infrequently and for short periods of time occurring in the passes. The fact that they were heard in 4 different winters supports the hypothesis that they might be using this as a migratory corridor. In contrast, looking at the UMNAC data, you see a different pattern, which seems to suggest that maybe they're using this a little bit more frequently and that it might be more of a summer habitat. But the detections in 2 different years in November and January suggest that they might also be using this as a migratory route. So these stars, the red stars, just illustrate where right whales were heard, and I overlap that with vessel traffic just to illustrate that they occur where we like to occur, and they are extending into the northern Bering. But this is, a tiny swath of their old range that they used to occupy, and there's a whole lot of ocean that they could still be. I think very interesting would be east and west of where we're sampling, which is something that I think Jess is going to touch on a little bit. And with that, I'll turn it over to Jess.
Great. Great, thanks. Dana just gave a really great summary about right whales, about why they are so critically endangered, and about our long-term mooring work. So I'm going to be talking about some of the surveys we've been doing, some other research that we've been doing with these animals, and then hopefully give you a little bit of a status update. So in recent dedicated surveys, there has been no dedicated survey in the southeastern Bering Sea since 2010. However, we have had a few surveys in the Gulf of Alaska since then. And I should say most of you are probably familiar with the results from the right whale program, the Priest survey that was from 2007 to 2010. So I'm going to be focusing on later years after that. So in the Gulf in 2011 and 12, this is a map of the areas that the IWC Power Cruise has surveyed. And you can see they did survey in kind of the northern Gulf of Alaska. There was one right whale sighting, but I'm going to get back to that, so hold on to that. And then in 2013, we had a survey in the Gulf of Alaska, a line transect survey called the Gulls Survey. All the dots here are sonobuoy deployments, and then I'm going to zoom in on the important part. The white circles, wait a minute, what happened? There it is. So the white circles there you can see are right whale detections. We did have 2 acoustic localizations on right whales, but unfortunately it was in the middle of humpback soup, and we never sighted the animals. Fairly similar story. 2 years later, in 2015, we were participating in the collaborative large whale survey with some funding from the Marine Mammal Commission. Thank you. And so these are our track lines here. These are some of the sonobuoy detections for the different species, but again, focusing on the important area. We did again have some right whale detections in that same area, right near Barnabas Trough, right off Kodiak. And we did have 2 different localizations, well, 4 different localizations on 2 different days. But again, we were in humpback whale soup, never got a sighting. So I've mentioned that we haven't had any dedicated funding in the southeastern Bering Sea since 2010. However, as Dana mentioned, we do have moorings deployed all throughout the eastern Bering Sea shelf. And every year when we go out to sea to retrieve and redeploy these moorings, we do opportunistic sonobuoy monitoring. And this plot here shows all of our sonobuoy detections of right whales as we transit to and from those mooring locations. And so you can see we do have a fair bit of coverage right in that one zone. We know right whales are there every summer. However, there has been very little effort in areas outside of this one zone. And so because of that, we do tend to rely very heavily on other surveys, other platforms, opportunistic sightings. And one of these platforms that we have been using lately is the sail drone. You guys heard about this a couple days ago with Jeremy and Carrie's work. So it's an autonomous sailing vessel that you can control remotely. It is filled with a suite of oceanographic instrumentation as well as fish echo sounders. And with the help of Marine Mammal Commission funding, we were able to also put a passive acoustic recorder on the sail drone to monitor for right whales. And you can see that in the photo in the upper right-hand corner. And so this instrument was set to sample continuously throughout the entire deployment. And in 2016, this map shows the track line taken by 2 sail drones. We had 2 boats going simultaneously. And you can see there was quite a bit of coverage, both in the the western portion of the habitat, but also further west, where we don't normally get to sample. Unfortunately, it was not unexpected, but the platform noise was pretty considerable with the water hitting the hull of the sail drone. So aside from a few random killer whale detections, we weren't really able to detect any large baleen whales. But then in 2017, they redesigned the sail drone. They removed the outrigger portion, they lengthened the hull, they filled it with a little bit of foam, and after some test trials, we decided to redeploy the Akusan. The results were a lot better. So we only had 1 boat running in the Bering, and this is the track line here. So the noise was reduced. It doesn't quite look like it in this photo, but I assure you it is. We did get quite a few killer whales. You can see some calls, hopefully, in the boxes outlined there. But more importantly, we did detect what we think are 3 probable right whale upcalls. So the first is in the critical habitat in late July, and I will outline right there if you saw that little red upsweep. We think we have an upcall there. Another one north of the critical habitat and outlined right there. And then the third, and what I think is the most convincing, is late August, again in the right whale critical habitat. You can see, it's hard to see probably for you guys back there. Unfortunately, though, there were no other right whale calls around this, so we cannot definitively say, yes, this was a right whale. However, we are fairly confident that we did. So this is still a platform. We are redeploying again. We're working still on quieting it down. In fact, we are hopefully about to do a trial in which we actually tow the recorder behind the sail drone. That would get the instrument off of the keel, off of the platform itself, and hopefully reduce a lot of that platform noise. So we're really excited to see these trials and see what comes of it. But then, in 2017, the IWC is systematically going to be surveying the Bering Sea from 2017 to 2019. Last year, we surveyed the eastern portion. You can see the track lines there in black. And because they were transiting right through the critical habitat, Phil convinced them that we need to have acoustics on board so that we can search for right whales. So I went on board with directional sonobuoys. You can see them here. They transmit in real time back to the vessel, any calling marine mammal that's in the area. So if you hear a species of interest, you can deploy 2 or more at once, and then you can actually localize on the calling animal and direct the vessel to them. And so we were successful. We found right whales. So this map shows the acoustic detections as well as the sighting locations in the stars. Quite a few were within the critical habitat. However, there was a big group that was to the east of the habitat, about 60 miles to the east inside Bristol Bay. And I'm really curious about this one. There isn't a lot of effort in this area. However, the aerial surveys from 2008 and 2009 did have coverage in Bristol Bay and did not find any right whales in this area. So I'm curious to see if this was an anomalous year or if this is an area that they frequent regularly, but we don't know it because there's a lack of effort. So breaking down these sightings, we actually had 12 unique right whales photographed on the power cruise. Of these, there were 8 known individuals, and from these known individuals, 4 were males. So we had 4 new individuals. 2 have been confirmed as new. The other 2 are assumed, but we don't either have the full suite of photographs or the quality isn't quite good enough to match. We did get 3 biopsy samples, and they were all from individuals of an unknown sex, which is really exciting. But most importantly, this animal right here is a juvenile, estimated to be between 1.5 and 4 years. And we did obtain a biopsy sample from this individual. We're still waiting on those results, but fingers crossed, Phoenix turns out to be a female. So what about other opportunistic sightings? Well, in 2011, there was one sighting in the Gulf of Alaska during a seismic survey. However, the photos were not of good enough quality to allow us to match that individual. I did mention the power survey earlier, how they surveyed the northern Gulf. There was one sighting in 2012 of a right whale. We were not able to obtain biopsy samples. They did not have the permit for it that year. However, this animal was confirmed to be a new individual. Sex was unknown. And then in 2013, you probably know, there were 2 different right whales sighted off British Columbia. The first in June was a female, was thought to be a subadult based on body size. And then later that year, in October, there was another individual, an adult that was sighted, this one with obvious entanglement scars. And both of these individuals were confirmed to be new animals. And then most recently, Dana mentioned sightings in Southern California. In mid-April, there was one animal sighted off La Jolla. Unfortunately, the photos were not of a good enough quality to match. However, about 3 weeks later, another right whale, a different animal, was sighted in the Channel Islands. And these photos were beautiful and allowed us to confirm that this was, in fact, another new animal. So you're thinking, great, we have all these new animals. What does that mean? Well, we still only have 24 definite animals in our catalog. And unfortunately, only 12 of these have been resighted. Only 5 of them have been resighted more than 3 times. And our star animal, the one that we see all the time, has only been sighted 8 times in the past 2 decades. So unfortunately, we have a very low resight rate, and we kind of rely on these extralimital sightings. And so because of that, we don't really have any updated population estimates, nothing new from the estimate that Paul Wade and his colleagues came up with. So our best guess is still around 30 animals. However, I think it's really encouraging that we have seen a lot of new individuals lately, especially the 2 juveniles. So I think it really further highlights the need for funding for these dedicated surveys. So there is also a really nice summary of this. The regional office just put out the 5-year status review for the North Pacific right whale. You can find it on the NOAA's website, so I definitely encourage you to look at that. It has all of this information plus a lot extra information. It's a really great resource. And I just want to wrap up with what we have coming up. So we do have our usual opportunistic monitoring surveys where we'll be going back and forth in between our moorings. So we will have our opportunistic sonobuoy deployments. The sail drone will be sailing again. However, this will be very Chukchi focused, so it'll basically just beeline it from Dutch Harbor north to the Chukchi. But we will have the Akusan deployed on that. And then the power crews will be sailing again in 2018 this summer. We will be doing the central bearing, so the green and yellow portions. You can see the track lines there. And we will have acoustics on board. I will be deploying our sonobuoys again, so I'm really excited for this cruise to see what we hear, what we find, if we do happen to find a right whale in that central bearing, and see if we can match it to either our catalog or the Russian catalog. Maybe start answering some of these questions and filling in some of these gaps. So that is all I have, 2.5 minutes to spare. And I think we're waiting on questions until the end, correct?
Yeah, if we have time.
Great. Thank you very much.
Thank you, Jessica and Dana. And we will wait, see if we have any time at the end for questions. And our next talk is on Cook Inlet belugas. And Paul Wade is going to start with a research update. And then Manolo Castellotti is going to talk about some of the acoustic work. Paul.
Thank you, Robert. Thanks for the invitation to talk to you again. But this is actually my talk as opposed to Phil's talk I gave the other day. A year and a half ago, I was asked to take over our Cook Inlet Beluga research project because Rod Hobbs retired. And that happened to be right at the same time that some funds were being made available for Cook Inlet Beluga research that were reprogrammed from pinniped work under the Species in the Spotlight initiative that our agency's been doing to spotlight some endangered species that are at great risk of extinction. So I've long known that it's better to be lucky than good, so I was quite lucky to come along when there was some new funding available. And we have started a lot of new research. I'm going to give you kind of a whirlwind tour through that, so hold on to your hats. So not much in results. I'm just going to be telling you about some of the new projects we've started. So to start stepping back a little bit, what did we have going on before we got this new pot of money to be able to use? At the Marine Mammal Lab, we were pretty much down to just doing our biennial aerial abundance surveys. And this is the survey that has documented the trend in the population that you see in the figure there from our latest stock assessment report. And it documented the severe decline that happened during the period of overhunting, and then has documented that for almost 2 decades, the population's essentially been stable or even slightly declining. And you can see our latest estimate from 2016 on there was 328 whales. So small population, highly endangered. And this is the big puzzle. We expected that it would be recovering by now, and we've seen no sign of recovery. There are other projects that have been going on, though, outside of the Marine Mammal Lab. Tamara McGuire's had a boat-based photo ID project for over a decade now. There was a Section 6 grant to Bob Small and Manolo doing acoustic work, and they did some really cool stuff there. And then Mandy Keogh has been doing stable isotope work on teeth. It's really interesting. But our lab did not have much more going on other than a few other small projects that Kim Sheldon was doing, for example. So to take a step back and think about, okay, what are we trying to study here? Why is the population not recovering? Excuse me. So, of course, we want to try and look at approximate causes, which hasn't really been done yet, and to look at, are they producing enough calves? Is their survival rate high enough? What's causing the lack of recovery? Or are both things going on? But, of course, we also want to get to potential causes, ultimate causes. And I've put down some of the things we're concerned about. And I don't have time to go through all these, but to point out the pollution thing, there's only primary treatment of sewage in Anchorage, and the whales travel right through the outfall area of that, so that's all been an area of concern for us. As Manolo will undoubtedly mention, there's a lot of noise and disturbance from shipping traffic and pile driving at the port. And we've had some concerns about prey. In particular, there have been declines in Chinook salmon runs in Cook Inlet, and including something that kind of took us by surprise, that there's some closures in the Susitna River going on this year for the first time, which is one of the prime feeding areas for the whales. And then also the vets have been telling us they've been seeing a few birth defects and necropsies. It's just a small number. I'm not saying that this is really happening, but it does give us concern about whether inbreeding is going on, some problems from inbreeding is going on. And then Randy and Reeves and I have long thought about this issue of social disruption that goes on in social odontocetes from overhunting and a severe decline, and wonder if that's something that could be preventing the recovery of the population as well. So these are some of the things we worry about. So now, what are we doing? So with this Species in the Spotlight money, we're doing a lot of new things. At latest count, I think we have maybe 17 to 19 new projects, or at least analyses. It's a little bit overwhelming, actually. And I've kind of put them in the various categories here, and I'm going to give you a quick run through all these, although Manolo is going to talk about the new acoustics work we're doing. So first of all, we are doing some new analyses. Charlotte Boyd has taken a very thorough look at how we estimate group size from the aerial survey data, and has come up with a new Bayesian group size estimation method that we think is way better than what we were doing before. So that's good. We've also undertaken a thorough review of how we actually conduct those surveys, the design, and I can't get into the details here, but we are going to experiment with doing a different design this summer that we think would be better. We're still going to run a number of days with the old method, so we'll be doing both this summer, but that's something we're looking into just to make sure we're doing the best job we can on those surveys. We also have a couple new modeling projects going on, both individual-based models. We have got some funding for a postdoc, Erin Jacobson, and she's doing an IBM model for stock assessment and essentially a population viability extinction risk as well. And then Charlotte Boyd again has got some funding from a Section 6 grant to ADF&G, working on trying to estimate vital rates, working with Tamara McGuire's photo ID catalog, and looking at what kind of sample sizes you need to get good estimates of fecundity and survival. And then we're hoping that Charlotte will eventually be able to work on adapt off those with an individual-based model that will incorporate physiology and prey year-round to sort of look and see, are there bottlenecks throughout the year, similar to what's been done with southern resident killer whales. Okay. Another big project that started 2 years ago, and I took it over last year, was to do biopsy sampling from boats in the inlet. And in the first 2 years of that, we've got 18 samples, and we're doing a lot with that. I'm going to run through in 2 slides all the different projects we have going on with that. First of all, Kim Parsons, who you heard from earlier today, is doing SNPs genotyping to do genetic IDs of the individuals, and we're hoping with enough sample sizes she'll be able to look at kinship studies and try and investigate inbreeding. Also, Terry Rolls has funded a beluga genome project. We're going to use samples from both Cook Inlet and Bristol Bay. This will supplement a recently published beluga genome from, I believe it was a Canadian population, so we're excited about that. Also, Scott Baker and I got an MPRB funding to do epigenetic aging, and Scott was able to hire Eleanor Boers as the postdoc to do that work down at Hatfield, and we're also partnering with Simon Jarman and Steve Horvath, who are kind of heavyweights in the epigenetic aging. So this should allow us to estimate the age of the whales from a biopsy sample, so we're very excited about that. We're also having Gina Ilatalo at the Northwest Center do stable isotopes to look at prey, and we're hoping to have enough money to try and do fatty acid analysis as well. And then on the second side, I've sort of grouped a lot of things that are sort of having to do with kind of health and things like that. Gina's also going to be running contaminant analysis. A long time ago, they ran legacy contaminants in this population, and the levels were actually quite low. So we're trying to focus on emerging pollutants, potential endocrine disruptors, and that kind of thing. Again, pointing back to the sewage that's coming into Cook Inlet from Anchorage. And Verena Gill at the Alaska region has completed a parallel study where they took water samples and fish samples from the Upper Cook Inlet and ran contaminant analysis on that. So we have that as a parallel study to compare to. And then a couple other studies that are funded externally that we were providing tissue samples for. Amy April and Amy Van Sice, who's actually here today from Hui, are doing a skin microbiome analysis, and we're providing tissue for that. Amy April has previously shown that the microbiomes, baleen whales can show changes between healthy and compromised whales, so it has the potential to be sort of an individual health index. So we're going to investigate that, and they're going to try and compare it to, say, other populations like Bristol Bay. Similarly, we're providing tissue to Tracy Romano at the Mystic Aquarium. They've been doing some RNA transcriptomics work to look at gene expression, particularly immune response, and they have samples from Bristol Bay and I believe Point Lay as well. Right, Robert? Yes, so those are a couple reference populations that we consider healthy that we can be comparing Cook Inlet to and look and see if there are any differences in gene expression. So that's exciting as well. And then also we're sending the blubber samples to Nick Keller to do hormone analysis for pregnancy and sexual maturity and stress. And I just have a tiny bit of results to show you, just to at least show you some result. The 5 blubber samples that were collected in 2016, cortisol at the top, progesterone at the bottom, and 4 were females on the right there. And you can see that 2 of the 4 females were pregnant. But kind of interesting was that one of the non-pregnant females had really elevated cortisol levels. But you can see it also had slightly elevated progesterone levels. And Nick pointed out that this is consistent with, but it doesn't prove this, but it's consistent with perhaps a terminated pregnancy caused by stress. So it's going to be really interesting as we get more of these samples to see, does this kind of thing show up repeatedly? Because this is just one whale right now, but we'll see what happens. And also, Nick showed that the male was not pregnant, so that was good. And then the other big project I started was to do photogrammetry work using hexacopters. You're all familiar with the work John Durbin's been doing with killer whales and baleen whales, and we're collaborating with him and his group. He sent up Hollis Europe and Jacob Barbaro to be the flight team, and they did a fantastic job last summer, and they're coming back up this summer. And we're also partnering with ADF&G to provide the boat, the platform that we work off. So I'm going to run you through that real quick. We using this very cool drone, which is essentially a flying camera, and most importantly, it has a laser altimeter on it so that we can do photogrammetry work and turn pixels into absolute measurements because we have a precise altitude. We've been primarily working in those shaded areas in the Susitna Delta and Eagle Bay, which are prime foraging habitat. We got some lovely photos like this, and this points out that in Cook Inlet we can't see through the water. It's really muddy, so we're having to work with just the part of the whale that's outside the water, so we can't do kind of the body condition work that John does with killer whales, unfortunately. We may look at body condition in the future, trying to look at depression behind the blowhole or something like that, but primarily we're just trying to measure the lengths of whales using the blowhole to the dorsal ridge here that I'm showing you with the red arrows, because this should correlate well with overall length. And most importantly, we think we ought to be able to identify calves from this, just as being the small guys. And we're working with people like Robert and others who can collect paired measurement data sets of us, of that blowhole to dorsal ridge measurement paired with a total length. And then we hope to produce out of that an annual calf production index. That's why we're doing the survey in August, kind of late in the year, once the calves have already been dropped. And really, we're trying to build a full sample of age structure from the population that we can use in model fitting. And we can translate it into age classes using the length age curve we have from stranded animals that have had a tooth aged. So we have a decent data set of that. And then we're also hoping to be able to connect these whales through to the photo ID catalog that Tamara has, and that's actually worked out really well. While they were flying the hexacopter, you can see here I was taking oblique photos at the same time to help us match into her oblique photo catalog, and it actually worked out really well. So we spent 10 days on the water in August. We did a bunch of flights, lots of hours of flight time, and took billions and billions of photographs. And Christy Sims and Janice Waite in our group have been doing the processing of all those photos. And importantly, they've been matching hexacopter photos to oblique photos that I was shooting at the same time. And it's worked out really well, it turns out. I'm just going to run you through quickly, showing matches they've come up with between the oblique photo I shot and the hexacopter photo. So we've shown that we can match up the hexacopter photos. They're high enough quality. So this is really nice. So now we can link in all our hexacopter photos that we're going to try and measure. We can link them into Tamara's 10-year photo ID catalog. And moreover, we can link her right side and left sides, which are separate catalogs. She only has a few of those linked. As Tamara's shown in the past, we're also seeing the whales that were tagged way back when. You can see the scars from the spider tags that show up on these whales, that some of them are still out there swimming around, and we caught that one. So the preliminary numbers are kind of gigantic. We were very successful. These numbers will go down once we do photo quality grading, but we have over 900 whales identified. That includes many repeat individuals across all these encounters. Over 500 are suitable for a measurement. About half of them, 460, are matched to an oblique photo. So we've got a tremendous data set that we should be able to do a lot with. So we're pretty excited about that. And then just real quick, it does give us another view of the animal for such things as this kind of funny skin condition that we typically see in young whales that the vets are interested in. And then also it gives us a better view of some of the injuries that we see in some of the animals. This incredible deep gash that's healed on this whale, but it's a female and it had a calf, so kind of remarkable. So that's a quick run through what we're doing, and thank you very much. And yeah, we're going to go to Manolo, right? Thank you.
Thank you, Paul, and Manolo will give us a presentation on another project funded by the Marine Mammal Commission on acoustics monitoring.
Yeah, thank you. So this is a quick, even quicker review than the one that Paul made. I'm going to be focusing on the acoustic work we did in Cook Inlet. So I have a list of co-authors here, but there is plenty of collaborators that I don't have the time to really go through them, but this is a large team effort. And so everything started in 2008. Actually, Shannon was probably the first person who proposed doing acoustics in Cook Inlet, and I think many people thought that it was impossible to do that. And so we basically tried our best to convince them and prove that we could do passive acoustic monitoring in Cook Inlet. So the first question for this project was to figure out if we could maintain moorings with acoustic recorders in a location that is mostly like a river more than an estuary. So we have over 33 feet of tide fluctuation, and that brings more than 11 knots, or around 11 knots of currents. That current is moving the sediment. Well, here you can see those pictures of the tidal fluctuation, which in some areas we have bow tides. There is 11 knots with the currents that is moving, basically moving the sediment on the bottom and having sediment such as sand, gravel, and even rocks rolling around in each tidal cycle that could destroy any mooring deployed in that area. There is glacial silt in some areas that has over 3 feet of sedimentation per month, and every summer there is people stuck in this type of mud, which is a very tricky and dangerous place to wander. There is also lots of vegetative debris coming out from the river, so the glacial silt comes out of the rivers, but also lots of branches and logs that are rolling down in the bottom, so the instruments get entangled, so it's very risky and very difficult to keep instruments in place. And of course, we have winter ice for half of the inlet, basically through the winter time. So all these factors make passive acoustic monitoring very challenging, and so we had to come up with very creative ways to develop our moorings and keep them in place. So basically, there you go. So most of the information we have from Cook Inlet belugas is mainly in summertime. So most of the aerial surveys have been done in summer, mainly in June. And so one of the objectives with this project was to try to cover the rest of the periods, October or November, fall, winter, and spring, and trying to make year-round monitoring effort in the area. Also, because we are recording sound, we also could hear other species such as killer whales and harbor porpoises, which are also common in the inlet. And obviously, we were also interested in looking at anthropogenic noise. So the same instrument could collect all the information for these 3 species together with the noise in their habitat. So we deployed instruments in 13 different locations. This is, again, the previous project that ended in 2013, which is the baseline for the new project that we're running now. And so you can see that our creativity with the design of our moorings, we had to build different peculiar configurations to make sure the instruments would survive the inlet, because if the moorings are linear, they would get entangled with all of the debris. They would be hit by ice in winter, and if we use very heavy anchors to keep them in place for the currents, they would sink in the mud. So we had to find a fine balance between all these elements to have instruments that could resist 6 or 8 months in the inlet without moving much from their location. And so the type of results we collected, and you can see several years of data here, this is just an example in Beluga River, are giving us a clearer understanding of seasonal presence in each of the locations we had mooring. So in this case, Beluga River, the west side of the big Susitna Delta, you can see a peak of beluga presence in June, July, when the first fish runs are happening in those rivers. And then again, this area is important between November and April, so it's a winter area for these animals. And you can see the consistency or inconsistency between years in these data sets. Very quickly, this is a quick summary of the acoustic effort we did in this project. So the circle radius means how much acoustic effort we had in each location. The number inside those circles is the number of acoustic positive hours, meaning an hour with acoustic detections of belugas. So you can see the left panel shows the summer information or the summer distribution, and the right panel is the winter distribution. So summer animals are highly concentrated in the upper inlet where there is most of the big important salmon runs, and that's where we know they concentrate for feeding. And in winter they disperse. You see we have more detections in the mid and lower inlet, but still they're still using very intensely the upper inlet, which is something that we learned with this acoustic that I was not expected to have belugas in both Nick Arm and Kern Point and the areas in the north end of the inlet. So we also found that with the acoustic data we could look at feeding behavior. We were able to distinguish feeding click trains from their echolocation, which are typical of chasing and capture phases that have been well described in porpoises and other odontocysts or dolphins, and little information on belugas. So what we did is we piggybacked in the project in Bristol Bay, where there is a big group coordinated by Fish and Game and our laboratory to capture belugas temporarily and then collect lots of samples. So that was a perfect opportunity to put DTAGs, which are suction cap instruments that record sound and movement and dive and depth information. So we put those DTAGs, satellite tags on these whales too, and then a stomach peel, which is a sensor that gives us information of when the animal is ingesting prey, because the temperature in the stomach gets lower when they eat cold fish, basically. So we instrumented a few belugas profiting those captures to look at feeding behavior. And basically, I'll see if I can play this file. Basically, we were able to find very clear sequences that correspond to the capture phase of the changing of these individuals. And let me play the spectrum you see down here. Basically, this is time. It's a few seconds, like 10, 15 seconds, and frequency from lower to higher frequency. Those vertical lines are each click, each signal the whale emits and listens for the echo to understand where the prey is. So it's a biosonar system. So you'll see that sequence, how the impulse interval is getting shorter by approaching the target. And then in this particular case, the fish is able to escape, the whale goes back to the prey, and so those intervals get closer, and then the fish turns around or is able to escape again, and so those intervals get longer. And by the end, the intervals get very, very close until the animal is captured. So let me play this, see if you can hear that. We are here more or less. There's a decrease, an increase, and more increase. And the end is a crushing of the fish. So you can clearly see how the whale is chasing and catching a prey. And we know these are successful catch because we got a drop in temperature. So we have pretty accurate information about the acoustic behavior of the biosonar when they're chasing, capturing fish. So now we get that information. We basically measure acoustic parameters in this sequence here, the feeding buzz when it's very, very close to the fish and capture the fish. And basically, we look at the interval between impulses to define when those intervals are short enough to be translated as a feeding event. And so we use that information with the mooring data and Cook Inlet to look at feeding behavior. And that was the analysis part that has been funded by the Environment Commission, which was critical for us to really better understand feeding behavior in Cook Inlet. So this is a summary of our current understanding of feeding behavior. When does it happen in Cook Inlet? That's summarizing all the mooring locations. So it's just a quick synthesis. So the index in the vertical axis is feeding positive minutes. So each minute with at least one feeding buzz detected in our mooring. So you see that there is a huge amount. Do you see my cursor? Oh, no. Well, anyway, there's a peak in feeding behavior in summer, as we would expect, because of the salmon runs. But if we use the amount of acoustic effort, meaning when we detect belugas, how often in those detection periods the animals are feeding, basically when we correct the amount of feeding behavior by the amount of beluga presence in our acoustic data, we find pretty different results, and that's what you see here in this orange plot. Now it seems that instead of summer, the peak feeding season is the month of May, or spring, basically. So that's a relatively new type of results that we didn't expect. And so in May, we looked at what are the prey species they can find in those rivers, and there's mainly three. There is the smelt, And there is a eulachon, which is 2 fish that are coming into the rivers in that time of the year, and also small, the young juvenile salmon coming out from the river. So it seems that those 3 species may be really important for coquinlet belugas, and May or early spring might be a very important period for them to feed. You see that June, July, August is still important in the orange plot, but May has a much higher level of acoustic feeding behavior. So the new project that started last fall is following up on that. Now we are focusing in feeding locations. We're trying to understand where do they feed year-round, not just in summer, but what happens in winter, and also what happens in those areas. Is there much noise that potentially disturbs these animals from foraging? Because that could be one of the indirect impacts by noise for successful foraging on these individuals. And we know there is quite a bit of noise. I don't have time to come into this, but there is, we published a paper in potential displacement by noise in Cook Inlet, and there is a good report where we described how much noise, how loud, how often, which sources of noise happen in Cook Inlet. So based on this information, we know noise could be a problem, and we're trying to understand how much of a problem could that be in their feeding areas. And that's it. Acknowledgements for many people involved. Thank you.
Let's thank all of our presenters here from the last session again. Thank you guys very much. And please catch them later for your questions. I'd also like to thank Dee and Vicki and Mike for organizing this, and we'll turn it back over to the chairman.
Okay, thank you, Robert. Well, we're at a point where we've lost some of our CSA members already. Peter, just 1 second. Okay. I know during the session here, the last 3 days, during the sessions, we've had good discussion. There have been things that have come out during that discussion that staff, Peter, have been recording possible things we might consider doing and so forth. So we're developing a list of things like that, but there really isn't time to either present it or discuss it, which is a problem that Francis and I talked about a little bit ago. So I think at this point, might just ask if anybody has any thoughts on anything over the entire meeting or today that you might want to throw out in the little bit of time we have remaining. Jason, go ahead.
Figured out now that I'll always talk if I'm given the opportunity. Nobody else wants to. Just because we're at this last session, and it's fresh, I just wanted to say this was an incredibly informative and impressive group of talks. But I want to single out the work on the North Pacific right whales. I'm always astounded by the spectacular work that you guys do with the minimal resources that you have. You definitely make the most of what you have. And it's been for years, and I'm a SEAL guy, so this is all still kind of new to me, but I struggle to understand why there isn't more emphasis on this species and more resources put to it. I think I know some of the reasons, but I can't help but point out that there are 15 times more North Atlantic right whales, and there's a tremendous amount of attention and resources being poured into that problem, rightly so, but I think it really is a stark contrast with the resources that these folks have. So I just wanted to point that out, if it wasn't obvious.
Yeah, actually, thank you for pointing that out. We pointed that out on a visit to headquarters a while ago. I don't remember exactly how long ago, but we pretty much were told it's good that you guys are putting some money into it because we're not. Dennis, go ahead.
So at one of our recent annual meetings, I made a comment that it might be a good idea to gather lessons learned from the vaquita situation for the next time that happened. And we heard yesterday that maybe southern resident killer whales is going to be a situation like that. So I'm wondering whether one of the things we couldn't be discussing is applying the lessons learned from vaquita to southern resident killer whale.
Anybody want to comment or respond?
Could just say yes.
Yeah, I was going to say, you know, southern resident killer whales are one of the most well-studied populations of whales in the world. There's been dozens of graduate degrees that have been undertaken on that population, starting in the, you know, late 1970s, early 1980s, and what, you know, from my perspective, what's needed is, not that I'm a scientist and I definitely think that more science is always needed and there's lots of unanswered questions, but, you know, the lesson from Vaquita and Baji before that is that, you know, science should not necessarily be an excuse for delaying action. And so it seems to me in the case of southern resident killer whales, action's what's needed. And science can go along with that to help inform the action and do it in an iterative or adaptive way, but you know, we need to focus on action.
Sorry about that. Yeah, that's more or less a comment I made yesterday, so I agree with you wholeheartedly. Robert?
Thank you, Mr. Chairman. I just wanted to say very briefly that I really enjoyed this meeting, and thank the commissioners and the staff for scheduling time for discussion. That, to me, the discussions held at this meeting were better than we've ever had, were more engaging and insightful than we've ever had before, and I really appreciate it and hope that we can continue, and hope that a lot of kind of the to-do list can come out of some of those discussions.
Thanks for that, Robert. And in fact, the staff have done a tremendous job in a very short period of time of pulling together. I agree, a tremendous meeting. And unfortunately, the timing of our meetings these days have come at a difficult time, though I guess there's never a good time, and as you get into the fall, we're at the end of the fiscal year, so we have difficulties with travel if we're overlapping years. Problem is, budgets are not, you know, at a point at this time of year often where we know if we have the money to hold the meeting or not. So staff really deserve an incredible round of applause for what they've done, especially with staff turnovers as well. So thank you, staff, for all you've done to bring this to a successful meeting. Okay, on that, I'll just sort of say, one of the things Francis and I talked about just before the beginning of this last session was, and it would be good if we had an opportunity at the end of our meeting. I know everybody's, you know, probably fairly worn out at this point, and yet it'd be good if we could have some discussion. As certainly the CCSA know, I think we can't hold some of these discussions in a private meeting. It's got to be in a public meeting. So adding perhaps a couple hours at the end of this last day to pick out highlights to talk about and sort of think about what we might do. In some cases, make some decisions on the spot of what we might do where it's obvious, is something we might consider for our next meeting. But I think this format that we've settled on with panels and discussions on key issues has really become, I think, the highlight of, and the fact that we're filling 45 minutes of discussion, and could probably go an hour on many of these, I think is just great, and we'll continue with this as far as I'm concerned, and I don't suspect I'd see any resistance from my two colleagues here. With that, let me give the opportunity to the little bit of time that's left to Francis or Michael if they have anything that they would like to say as commissioners here.
Just another comment really to thank the staff for setting up a webinar that's actually not been obvious to people in the room, but it's actually been. Sam Simmons and Brady in the back here have been fielding questions from the public, and we've been emailing responses, and I think it's made this meeting a lot more accessible to people who couldn't travel. So the fact that it did appear so seamless means that they really did a good job. So thank you for that.
So reflecting back on some of the comments and my experience with on the killer whale panels, you have 9 assembled experts, and you ask them, try to give me a consensus on what are the actions that you recommend. And 3 of the 9 did do that, but the other 6 didn't. So I reflect on why that is, and in some respects it might be because, well, I'm a scientist and I have a certain area of expertise, and if I tell him I want him to focus on some other guy's expertise, then that means my funding will disappear. You know, maybe there is that involved. What I'm trying to get at is, how do we ferret out from people the kinds of advice that we need to do our job? If someone can answer that question for me, do so. And then the other thing is, we're a bunch of scientists, science-based, and but what we have to do is get agencies, both federal, state, local, and so on, to try to do something to change their behaviors to make possible the survival of some of these species. And that can be difficult. In the case of the governments, it goes back to the vaquita. Let's prevent a vaquita situation occurring again. But in the vaquita, at least you had a consensus of the scientists for a very long time of what the problem was. It was that Totoaba fishery and the bycatch. So everyone could focus on that. But what was missing there was the political will of that society to do what it had to do. And that is in a context of a particular kind of climate where corruption runs rampant. But anyway, the Mexican government did do a miraculous thing by stopping a fishery from happening for a while, even though corrupt individuals and so on helped prevent it from being effective. So I'm rambling on here because I'm trying to say to you, yes, it's easy to define the actions that ought to be taken, but then where do you go? How do you force that to occur? And it takes the political will of the public to agree to do that. And in the case of the southern resident killer whale, they have to say, well, how much are we willing to invest in the establishment of a hatchery system, or how much are we willing to bite the bullet and blow up all the dams and all these rivers? Or just what are the costs are we willing to bear as a society to make sure that this stock continues on? So again, if someone can tell me how to persuade the public to do something that we think is needed, please let me know. And also on the killer whale, the thing that struck me is how we're in these regime shifts due to climate change. And I was struck by, in the very beginning, we had those presentations about, well, here's the state of the ecosystem. And one of the experts there informed us, well, as far as we can tell, the climate is changing in the Pacific Northwest from a snow-driven system, snowpack-driven system, to a rainwater-driven system, meaning you're switching over to a very warm climate, and the kinds of situations, environmental conditions necessary for survival of salmon aren't going to be met anymore. So that's the long-range view. So that's the other thing that you have to take into account. There are short-range actions that can do something and maybe save the killer whale, but in the longer run, are they really going to persist if they depend upon Chinook salmon? And that whole system is, putting it in a polite sense, screwed. So in some regards, it's kind of hopeless. I'm sorry to leave it on that down note. That's where I got to on killer whales. And Dennis was asking me, why didn't I weigh in on the managers like I weighed in on the scientists? Because I started thinking about these things, and I sort of got kind of dispirited. And I said, screw it.
Thank you, Mike. I'm going to bring us back around like Francis did for me when I was perhaps on this similar set of comments. I think, for me, I don't think it is hopeless, especially some of the things we saw today. I was really impressed with the group, the working group that involved people representing stakeholders that we often see at very strong odds with one another in these kind of meetings, sitting together, sharing their time, working together towards a problem. I see it as optimistic. I see it as optimistic, actually, that the Marine Mammal Commission is still here to have its meetings, that we have our budget because Congress, and I think Congress is responding to the general public, and so I think there's reason to be optimistic, even though things happen slowly, and that's the real problem perhaps at times is we're faced with situations where, the vaquita being a great example, where you just can't do enough fast enough. So with that, I think I'd just say thanks to everyone who's still here, who's been part of the meeting, and we'll be back to this part of the country probably in something less than 10 years. Thank you all. Okay, do we actually have to vote to close the meeting? No. Okay, I guess I'm official. Meeting is now adjourned.